team. Today we're talking about replication. 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 So we've already talked in some detail about why two studies may render different results. But as we've presented things so far, it's kind of seemed binary, right? Studies replicate or they fail to replicate. But actually, evaluating whether the results of the second study support the original is not always straightforward. And in fact, even determining whether something constitutes a replication attempt can be a complicated question. For instance, let's say I come up with a delicious cookie recipe. Bake it, love it. Everybody says 5 stars. And then someone comes along and attempts to replicate my finding that the recipe renders delicious results. And they arrive at a different conclusion. I would argue that this replication attempt, this failure to replicate my results, doesn't actually undermine my original finding, right? So today we're going to talk about how we can evaluate replication attempts. What does it mean to call something a replication? What are the criteria by which we evaluate whether a study replicated or not? And how can we make the research that we are doing right now, the new studies that are being published now, more replicable? So the first distinction laid out in the articles that you read today has to do with the distinction between direct and conceptual replication. In direct replications, the researchers attempt to conduct a study in a manner as close to the original as possible. Conceptual replications, on the other hand, attempt to test the same fundamental idea behind the original study, but there may be big differences in how the variables are operationalized, the study design, participant populations, and so forth. In class, I'm going to assign you to teams and make you argue for the benefits of direct versus conceptual replications. A second important key to evaluating replications is that an individual replication attempt shouldn't actually be considered a success or a failure. It isn't as straightforward as that. We can't think of these as simple, yes, it replicates, or no, it doesn't replicate votes. A replication that renders a statistically significant result in line with the original may be considered a successful replication. But if the effect size of the replication is far smaller, that's somewhat weaker evidence than a larger effect would have been. Further, a study that fails to replicate an original finding, but deviated in some important ways, like my cookie recipe, isn't exactly a failure to replicate. So when we look at replication attempts, we shouldn't just do like a nose count and say, effect shows up in one study and it doesn't show up in 2 studies, it must not be true. The next key point to keep in mind is that studies need to be evaluated on more than just whether or not they agree with one another. That is, if we have 2 studies that are done in exactly the same way and render different results, it doesn't mean that the strength of the evidence that each study provides is necessarily equivalent. So for example, let's say the original study had a relatively small sample size and provided lots of opportunities for researcher degrees of freedom and reports a surprising and unexpected finding. Study 2 is a direct replication. It is done as similarly as possible, but it has a much larger sample size for more highly powered analyses, and it's pre-registered to ensure that the researchers are running the study exactly as specified and only conducting the specified analyses. If they do not replicate the original effect, what are we left with? I would argue that study 2, with a larger sample size and good evidence against p-hacking and harking, tends to provide stronger evidence than study 1. It's also important to remember that just because one study came first doesn't give it any special status. If study 2 had come first, if that were the original finding, would study 1 convince us that study 2 was wrong? So this time reversal heuristic can be useful when you're evaluating replication attempts. Being first doesn't necessarily mean it was done better or worse. Okay, a third thing to keep in mind when evaluating replication attempts is that the criteria of what constitutes a direct replication differs. So one of the studies that was part of the reproducibility project had to do with how sugar affects decision making. And the idea is that having energy depleted will lead to more reliance on cognitive heuristics, will change decision making. So the original study included more components than I'm going to talk about here, but the important point for our purposes, the setup is that participants get lemonade that has real sugar or fake sugar, Splenda, and then they're asked to make some kind of difficult decision. So in this case, the hard decision was evaluating apartments that varied on a number of criteria, including how far away they are from campus and how nice they are. So for instance, you could have a nice apartment that's a 20-minute drive away, or a crummier apartment that's a 5-minute drive. So in the original study, the researchers found that people who had real sugar seemed to use different kinds of decision-making processes than people who had fake sugar, in support of the hypotheses. The replication attempt did not find any difference between the real sugar group and the fake sugar group. But here's a critical difference. The original study was done at Florida State, which is a commuter school at which students regularly have to make this exact kind of decision about where they're going to live. Do you want to live somewhere that's better but 18 miles away, or worse but 10 miles away? And the replication attempt was done at UVA, which is a residential campus more like Carleton. So you, Carleton students, probably haven't ever had to make the decision about whether you're going to live 10 or 20 miles off campus, because that isn't an option that Carleton allows. So the key point of the study is that sugar affects making a difficult decision. But maybe that kind of decision, so maybe that kind of decision is hard and requires a lot of thought for someone at a commuter school who regularly has to make that kind of decision, but doesn't require the same amount of difficulty for someone who doesn't often think about that. Maybe you're taking it less seriously because that's a decision you've never made before. So it may be that a hard decision at a campus like UVA or Carleton would be something more like making a decision about room draw. So maybe this isn't actually a failure to replicate, it's a demonstration of the boundary conditions of this finding. We're going to talk in class more about how we determine whether or not a study is a failure to replicate or a boundary condition, and how we can determine whether hidden moderator variables are affecting the outcomes. So what that means is that when we ask the question, did it replicate, the answer often turns out to be kind of complicated. Although some people think that science is in the business of delivering clear, pat answers, the fact is, it is not. But very importantly, this doesn't mean that we can't evaluate the evidence for and against something. It doesn't mean that the fact that studies conflict doesn't mean that we can't ever know the truth. It means that science is hard, and figuring out complicated things about human psychology is complicated. But with a sharp set of eyes and knowing what to look for, we can evaluate the evidence for and against particular ideas. So what I want to end with today are some of the ideas that have been developed in the last few years to help resolve some of the uncertainty about replication attempts. The first of these is the advent of the Registered Replication Report. And as the name suggests, this is a registered report of replications. So in registered replication reports, a researcher or team of researchers submits a proposal to the editors and makes an argument for why a particular study should be replicated. It's cited a lot. Everyone thinks it's important. It's the foundation of this whole research that we're doing. And if they make a compelling case, they then develop a study protocol that includes details about exactly how they're going to implement it and what methods they're going to use. Whenever possible, the original author of the study that's being replicated signs off on the fact that the replication is being done in a way that the effect should be expected to emerge. Then the study is run the same way in multiple labs to attain large and diverse samples, and all of the data are pooled and analyzed. So registered replication reports are a really compelling way to run replications because they ensure ahead of time that the results of the study will be clearly interpretable. So none of this like making cookies with motor oil where after the fact we can say, yeah, you shouldn't have done it that way, right? Especially given that the original authors sign off on the methods, it isn't possible to say after the fact that there was some hidden moderator variable that changed the results in some critical way. Another proposed change to help combat confusion about replication is for researchers to be more explicit in new studies about the conditions that they expect their results to extend to. So when we do studies, we assume that our results will generalize. So I study human speech perception, which means I'm not interested in how Carleton students understand the 200 words that I happen to play for them in this study. I want to know things about humans and how they perceive all of the words. So I expect my results to generalize beyond just the sample that I have collected. So here's another example. If I find that the color red, seeing the color red, makes Carleton undergraduates act more impulsively, I'll write up a paper saying red makes people impulsive. I'm not saying that this exact shade of scarlet makes the 25 Carleton undergrads between the ages of 18 and 22 tested in the fall of 2020 act impulsive, as measured by this one measure of impulsivity. I'm saying red makes people act impulsively. So does that mean that any study using any shade of red and any measure of impulsivity should be expected to find the same results? Maybe, maybe not. But as a researcher, one thing that I can do is include in my paper a constraints on generality section that specifies exactly the conditions I think my results should extend to. So maybe I want to be clear that red causing impulsivity is something that's only likely to emerge for young people, given evidence that old people make decisions in different ways. Or maybe I want to be clear that this finding should only extend to really bright shades of red because those are the ones that have associations with impulsivity. Or maybe I want to specify that the red has to be present for a long time in order to activate those associations. So then, if a replication attempt comes along and tries to replicate my finding, and they show the color maroon to old people for a second, and they find a different result, I can say, yeah, because color and amount of exposure and population matter. So constraints on generality sections are really useful because they help encourage researchers not to overpromise or overextend their results. It also helps to motivate future research questions for those researchers or others. When I've written constraints on generality sections in my lab, my students and I have often said, would this extend to older adults? That's actually a really good question. What are the reasons that older adults might be processing information differently and so would show different effects? And so that can be a starting point for future research. It also helps to ensure that direct replications are sufficiently direct. So if other people want to replicate my work, this is my opportunity to tell them in the scientific record, as written in stone as you can get in science, these are the features that I think are particularly important. Make sure you follow these guidelines. So the lead author on the paper in which the constraints on generality sections was proposed is a researcher called Dan Simons. He also wrote the article that you read for today, advocating for the value of direct replications. Oh, he also pioneered registered replication reports and is the editor-in-chief of the journal that publishes them. And guess what? Yeah, he's a Carl, class of 91. He's also the invisible gorilla guy. Okay, so finally, one of the other ways to facilitate future replication attempts of new work is to include highly detailed method sections. So include all relevant information, and even better, share materials and protocols to ensure that future work knows exactly what you did. So in class, we're going to talk about some quote-unquote failures to replicate. We're going to talk about the benefits of direct versus conceptual replications, and we're going to discuss how to encourage more researchers to conduct replications of their own. Thanks for tuning in, and I'll see you in class.